Welcome to this explainer. Okay, let's jump straight into it. Picture this. You're a veterinarian and you've just been called out to a massive agricultural facility. Animals are just dropping dead. No prior warning, no obvious signs, and you have absolutely no idea why. Well, this is the very real, very high-pressure reality of facing explosive agricultural disease outbreaks. Today, we are going to unpack the high-stakes medical detective work required to stop a fast-moving, microscopic killer before it completely destroys a local economy and threatens our food supply. So, this brings us to the central mystery we need to solve. How exactly do you identify a microscopic killer before it wipes out an entire herd or flock? I mean, we're dealing with invisible threats that move incredibly fast. You can't just swab an animal, send it off to a standard laboratory, and wait around for an answer a week later. The lives of thousands of animals, and frankly, the livelihoods of farmers, depend on rapid, on-the-spot identification. Let's meet our prime suspects, the Pasteurella safe family of bacteria. Now, what's really fascinating here is that these bacteria are actually gram-negative commensals. That's just a fancy scientific way of saying they usually live completely harmlessly in the mucous linings of the upper respiratory tract of healthy animals. They're just freeloading, hanging out there. But under the right, or actually, scratch that, under the wrong conditions, they transform from quiet passengers into incredibly deadly pathogens, invading tissues and triggering massive systemic infections. To see how this threat builds, let's compare the two major bad actors in this family, Pasteurella and Mannheimia. Now, they're closely related. In fact, Mannheimia haemolytica actually used to be classified as a Pasteurella, but they have very different targets. Pasteurella has a massively wide host range, causing terrible diseases like fowl cholera in birds and hemorrhagic septicemia in cattle and buffaloes. Mannheimia, on the other hand, is mostly restricted to ruminants, like sheep and cows, where it causes severe necrotizing pneumonia. So scientists and vets have to carefully differentiate between these two to stop an outbreak in its tracks. Look, the absolute most crucial factor here is time. Rapid diagnosis is literally critical. If you take a sample, put it on a blood agar plate, and incubate it at 37 degrees Celsius, you're looking at 24 to 48 hours just to see any bacterial growth. In the middle of a rapidly fatal outbreak, waiting a day or two for a lab culture simply isn't an option. By the time the lab calls you back, you could have lost the entire herd. We need an immediate, on-site answer. And this is where our core focus comes in. How do we get that immediate answer? Through a really clever phenomenon called bipolar staining. When these bacteria are smeared onto a glass slide and treated with a specific dye, a Giemsa stain, they exhibit a highly distinct visual pattern. The ends of the tiny rod-shaped bacteria absorb the stain intensely, while the middle stays lighter. Under a microscope, they look exactly like tiny little safety pins. It's almost cute until you remember how deadly they are. This bipolar staining is the absolute game changer. It's the critical forensic fingerprint that tells a vet exactly what they're dealing with right then and there. Let's break down this practical field procedure that's out there saving lives. It's a super fast, three-step process. Step 1. The vet collects a blood or tissue smear from an infected or recently dead animal. Step 2. They apply that Giemsa stain directly to the smear. Step 3. They look under the microscope and hunt for those safety pin organisms. Spotting that bipolar staining fingerprint at the scene confirms the presence of these deadly bacteria instantly. Boom. You've just completely bypassed the need for days of laboratory incubation. If we check out the epidemiological threat board, we can see exactly which bacterial serotypes are causing these nightmare scenarios. Pasteurella multocida type A, that's taking down poultry with fowl cholera. Types B and E are out there devastating cattle and buffaloes with hemorrhagic septicemia. And meanwhile, Mannheimia haemolytica is causing bovine pneumonic pasteurellosis, which you might know better as shipping fever, in ruminants. Each serotype has a specific target, sure, but they all share that exact same telltale bipolar staining signature. All right, let's open our first case file, hemorrhagic septicemia. This is an acute, incredibly fast, rapidly fatal disease that primarily targets cattle and buffaloes, with buffaloes actually being the more susceptible of the two. It is a massive economic threat in regions where the disease is naturally entrenched, like parts of Asia, the Middle East, and Africa. And the scary thing is, it doesn't just happen out of nowhere. It's highly opportunistic. It's often triggered by severe environmental stress. Think monsoon rains, poor body condition, or overwork in draft animals. The timeline of this disease is, frankly, terrifying. It has the super short incubation period of just 2 to 4 days.
Once it takes hold, animals suffer a sudden onset of high fever and severe respiratory distress. A major hallmark to look out for is characteristic edema, massive swelling right in the throat or laryngeal region, which can even spread down to the brisket. Very quickly, the animal becomes recumbent, meaning they lie down and can't get back up. And they die quickly from endotoxemia, a massive release of toxins into the blood. It moves so fast that sometimes sudden death occurs without anyone noticing any prior warning signs at all. And this number right here is why we are talking about this. 100. When hemorrhagic septicemia strikes, mortality rates are usually over 50%, but they can easily approach a staggering 100%. If this bug gets into a stressed, susceptible herd, it can wipe out virtually every single animal in a matter of days. So how on earth do we stop a disease that kills nearly 100% of its victims in days? Well, we rely on our forensic clue. By taking blood smears from a recently dead animal and applying that Giemsa stain, vets can immediately reveal large numbers of those bipolar staining organisms. Finding that safety pin fingerprint is the silver bullet. It allows the vet to instantly confirm the diagnosis and immediately start saving the rest of the herd before the bacteria can claim even more victims. Let's shift our investigation over to a second major threat. Case file number 2, fowl cholera. This one is caused by Pasteurella Multocida, capsular type A. It is a highly contagious disease that aggressively attacks both domestic poultry and wild birds, with turkeys actually being particularly susceptible. Just like in cattle, this bacterium can live quietly in a flock until the conditions are just right, and then it strikes with absolutely devastating force. When fowl cholera erupts, it acts as an acute, fatal septicemia, and it spreads through a flock like wildfire. The bacteria invade the bloodstream, causing severe internal hemorrhages on the outer linings of the organs and massive fluid accumulation in the body cavities of the birds. Just like hemorrhagic septicemia, it is incredibly fast-moving and lethal. Rapid identification is literally the only thing standing between a farmer and total economic devastation. And once again, guess where the diagnostic breakthrough comes from? Yep, the microscope. In the acute septicemic form of fowl cholera, blood smears will show numerous characteristic bipolar staining organisms. Seeing that distinct visual fingerprint in the blood of affected birds gives veterinarians the instant microscopic verdict they desperately need. It proves that this bipolar stain isn't just a neat laboratory trick. It is a vital, universal tool for identifying this deadly family of bacteria across completely different animal species. So why does catching this visual fingerprint so quickly matter? Because rapid diagnosis quite literally saves lives. Identifying the bacteria under the microscope allows veterinarians to immediately administer life-saving antibiotic therapies, like tetracyclines or sulfonamides, during the early fever stages when they are actually going to be effective. It means medication can be instantly added to the flock's feed or water. It also allows authorities to enact strict isolation or, in regions where the disease is exotic, slaughter policies to completely contain the outbreak before it spreads to neighboring farms. But if we step back for a second to look at the bigger picture, we can't just talk about the bacteria without talking about why they strike in the first place. Remember, these bugs are often harmless commensals. The true catalysts for these explosive outbreaks are environmental stress factors. We're talking things like overcrowding, sudden chilling, drastic weather changes like monsoon rains, and the severe stress of transportation. These stressors weaken the animal's immune systems, giving the latent bacteria the perfect window to invade the tissues and multiply. And this quote brilliantly illustrates the danger of modern agricultural logistics. To quote the source directly, explosive outbreaks of disease can occur if an active carrier is introduced into a stressed, susceptible population. Older animals are often latent carriers, hiding these bacteria deep down in the pockets of their tonsils. When they get stressed, they shed the bacteria in their nasal secretions. Bring just one of those stressed carriers into a new, vulnerable herd, you have literally just lit a match in a powder keg. Which leaves us with a pretty critical thought to ponder. As our global food supply chains demand that we transport animals more frequently and over much longer distances, we're inadvertently creating the exact stressful conditions that these bacteria thrive on. Thanks to the quick identification provided by bipolar staining, veterinarians out in the field have a fighting chance. But as we continue to shift and move susceptible populations all over the globe, how will we stay one step ahead in this high-stakes, evolutionary arms race against these opportunistic killers? It's a massive puzzle that agriculture and science are going to have to keep solving together.